Did you know that GSU is moving towards Zoom? Officially, it will be next semester, but they already have it this semester as well. So I think you, yeah, this is my pro account. I'm not using GSU yet. That will switch soon in a week or so. I think what you need to do, like go to Zoom, log in, then choose SSO, then write GSU-edu, something like that. Then you go through the GSU login and all the credentials, and then you should get your pro account. Sorry? What is it? What is it? Why? You scared her? What did you say? Something in Indian? Oh, oh, do you like it? Do you like in person or online? How many of you like online courses? In person? So everyone, 50-50, right? It's good to have some interaction, okay? Sometimes Zoom or online meetings good for collaborators. Like if you are collaborating from someone outside Georgia, you don't have to travel. Before COVID, it was really troubling to travel a lot for conferences, for universities, and so on. You need to prepare for the visa, to do all the booking, to take some time off from your school. So it's really large hassle to be handled. And not every travel really worth it. So sometimes really online meeting will solve the thing. But for teaching, I don't think it makes any sense to teach online. You will lose that sense of interaction. So you don't know whether the student is really understanding, whether he's bored, he didn't get the idea, right? So you're just like you're talking to yourself. You lose that feedback. Likewise, I think for the student. So yeah, you lose the chocolate, right? Yeah, so I made that promise that every time I will not go back home with any chocolate. So please try to participate, okay? Yeah, maybe we can wait a few minutes. How's it going so far with the course? I'm sure everyone already took the computer architecture class during undergrads. How many of you took that course? Okay, yeah, makes sense because it's a prerequisite, I assume. How about digital design? Okay. So what was the last lecture in computer architecture during undergrad? Is it about pipelining? How many of you studied pipeline or pipelining? Instruction pipelining. How you increase parallelism in the CPU side. Okay, so yeah, I'm thinking the last two lectures will be around instruction pipelining. So we already studied a lot about the cache, how to improve it, how to redesign it, what are the alternative options, right? And we moved to the memory system, especially the primary memory, which is DRAM device. We studied a lot of things about it, all the optimization techniques and so on, how to make it better, how to redesign it. But also we went through processing in memory. And processing in memory is not about only memory device, but it's about caches, storage, and all these. It's still considering as processing in memory. So probably we'll revisit the CPU. We didn't touch it yet. We didn't talk about it, right? We talked several times about the need to convert from C to assembly language to machine code, and then a little bit about ALU, the microarchitecture that we have over there, and so on. But we didn't cover it really in depth as we did with the caches and the memory. So probably we need to revisit the CPU, give you a lot of opportunities on how to improve it. Especially the pipelining is a really important topic. And then we may cover something related to LLMs or transformer architecture, how to make it better, how to optimize it. What is what are the trends in improving the LLM from the architecture point of view, okay? And then we will jump into the student presentation. It will be your turn to present to the class. You'll take over the stage, and you will show us your innovative way of summarizing or presenting a paper, okay? There is really no limit in how you present. In terms of ideas, in terms of topic, it's up to you to cover whatever you want. Of course, there is time limit, but in terms of the slide, the topic, the example, the tutorial you want to show, it's really up to you. So I will leave that side to you, how much in depth you go to cover the topic presented in the paper. Remember, the goal to present a paper is really not to present the paper itself. You are presenting a topic rather than the paper. So if the paper is talking about processing in memory, you want to teach us about processing in memory, okay? It's not like those people just woke up some crazy day, they come up with that idea, and this is the idea, this is the result, and that's it. This is really not how we present research paper, okay? It's not really interesting to know what the people did. It might be okay to get an idea that this work has been done. But you have to really show the insights, the hard work that all the way conveyed with that product, okay? So give me the challenges, the problem they start working on it, what the other people been working on, and so on. So we cover a lot of these things, a lot of these advices on the second lecture. So hopefully everything is clear to you. We already start individual meetings with some of you. Please make sure you schedule an appointment with us, me and Sapper, at least a month before your presentation date, okay? That is the first meeting. And then we should have at least 2 more meetings after that. So the first one should be a month apart. You could plan for 2 months apart, that's up to you, but don't make it less than a month, okay? This is where I will give you a lot of suggestions on how you start. So it will be better to give yourself, your group, really enough time to prepare well in advance, okay? That will help you to nail it, to make a perfect presentation. And if there is any missing point, you already have enough time to study about it, to read more, to prepare enough slides about it. Hopefully it will be clear as we start meeting. So please make sure you exploit this opportunity. I'm doing it just for your benefit, right? Okay, so today, hopefully it will be the last lecture on processing in memory. We already covered several papers, several topics, several innovative ways of how you could enable the processing near the data or where we store the data itself. Today, we'll continue with the complex operation. Last time, we discussed one topic about it. Do you remember what was it? Pluto, right? So yeah, Pluto was one of the ways to enable those complex operation inside the memory device. Remember this picture? This is really important. These days, we are not talking about the Neumann model. We start shifting the paradigm to something else. And that shift is really not defined yet. We don't know what will be in the future, in the next 5 years, 10 years down in the road. We have no idea about it. But we have a lot of ideas on how to improve certain applications. Which one is better? Which one will be the future? We don't know. Okay? It really depends on the application that we are targeting. If everyone is shifting into transformer-based application, then we could have better idea on what will be the future. Okay? But until today, I want you to be aware about all those different architecture. When we talk about intelligent software, what does it mean? Directly, you should know that the software that is aware about the hardware. Okay? Hardware-friendly software, the algorithm that exploit a lot of optimization from the compiler point of view. We talk about loop interchange, for example, blocking, and many more. Multi-threading could be one of them. There's a lot of things that you could exploit from the way you programmed your software. We talk also about AVX. You still remember it? AVX 512, this is available in Intel. Some other processor, they provide different variants of that. but with different instructions. So you have to be aware about the instruction set that you should use. Hopefully you remember what is AVX. SIMD instruction, which is, what does it stand for? Excellent. Single instruction, multiple data. What does it mean? Dividing. Excellent. So we divide the operands, or we collect all operands and we put them in one vector, and then we send that vector to the CPU, apply one operation on the entire vector. Okay? We divided among the operands that we have, and that division is really abstraction. So it's up to you. You want to have it 4 bits per operand, 8 bits, 16, it's up to you. Okay? Likewise, with the GPU architecture, a similar thing. So you want to have like register or full array of data, and then you distribute it among all the threads that we have in the GPU. Same concept. We also talk about fundamentally new storage architecture. This is not something really fixed. So every day we'll show, or we will go through really different, fundamentally different storage architecture. So we're not talking about larger capacity or faster memory. Yeah, that's the trend. Every year they are getting improved. But we are talking about here totally fundamentally different storage. Something that we never think about. It's not SSD anymore. It's not DRAM anymore. So we see the shift to the synthetic DNA, for example, right? That is what we are talking about. Really totally different medium of storage. We'll go back to this famous 5 forms of processing in memory. We cover a lot with over here and somewhere over here where we do PIM next to the memory controller, or PIM by leveraging the analog operation inside the DRAM. Okay, we didn't talk much about how we could add compute engine to the DRAM without touching the analog operation. We're not modifying the way that the DRAM works, okay? But we are enabling some small cores, could be FPGA cores, for example, inside the DIMM. And one famous example of this is the French startup company called UpMem, which is already acquired by another company this year. So I will leave that up to you. You will see a lot of material online covering a lot of things about UpMem architecture and how you program it. I was planning to cover some lectures on this and give you like really hands-on tutorial on how to program it. It's really similar to Python or C code. You just need to add special instruction where you move the data to the device, where you get the result from the device, and how many engine or how many compute engine you want to activate inside, and how you distribute the data and so on. But maybe we'll cover that in the next semester. We also have access to Ryzen AI servers. So if you're interested in doing anything about that, I can grant you access. This is from AMD. They already have the devices, very small devices. You can connect through USB to your laptop. I think the cost is about $200 or something. Real small devices. But we do have access to the huge cluster, so you can do unlimited compute over there. So if you are doing anything related to LLM, we can talk, okay? Today, we'll try to cover something related to 3D stacked memory. So we already complete many examples of the PIM inside the DRAM device. Today, we'll visit a new topic, which is related to 3D stacked memories. Okay? Maybe we will cover one paper about that, near storage processing, or we'll leave it up to you. I think there is one group. Probably there is one group covering a topic about that. But we may cover something else related to genomics in the next few weeks. We will discuss that. Hopefully you still remember row clone. This is a really important paper, how you can leverage the DRAM device that we saw several times. This is Ambit paper and an OR operation only. This is AMPIT with the NUT operation. And we talk about how to support the execution or complex operation in DRAM. We're still in DRAM device. We talk about Ploto. Hopefully you remember all the operation over there. So if not, I will quickly cover it just as a recall. So we have that problem as an example. You are looking for finding the second, first, second, and fourth prime numbers. And we discussed last lecture how we build that lookup table. Hopefully it's clear to you. So we have the index of the element. We have the index over there. We have the function. This is the input to our lookup table, and this is what we are supposed to get. So based on those indices, we are supposed to get the output that's stored in the lookup table. And again, we said this is mimicking as if we are doing the complex operation, but there is no computation happening here. Okay, that's why we're not adding any compute engine to the DRAM. So this is the problem again to this side. This is the DRAM organization. And we said we duplicate the lookup table into column wise just to leverage the parallelism enabled by the DRAM accesses. And now if we have the input vector over there, we remember we access the DRAM always row wise. We fetch one of them, we bring it, and now based on the column decoder that is attached to the input vector through the match logic, if anything matching the index of that row, then we fetch it into the column decoder. So we keep checking one by one. Are we in row 1? No, we are in row 0, so skip. Are we in row 0? Yes, so bring that element to the column decoder. Are we in row 0, row 1? No. Are we in row 3? No. So just ignore. Okay. So we just get one element because that row matching the index of the input vector that we are looking for. And really, when you check the result, what is the first prime number that has an index 0? It's basically 2, not 1. Remember, 1 is not a prime number. Okay. So if you repeat that to all rows, you'll get the output over there as if we are calculating the prime number, but we are not doing any calculation, basically just fetching the data over the DRAM itself. All right, that was just a quick visit to the Ploto, which is a very important topic. But remember, we talk about a lot of limitation, a lot of space for improving Ploto itself. And we said we see great benefit up to, I think, 16-bit query. So the number that we are looking for our query that we are trying to search inside the lookup table, at most 16-bit wide. After that, we start losing the bandwidth, start getting really slower. And we discuss why. So we already have all that kind of discussion about it in the previous lecture. Those kind of questions really are important in your student presentation. Okay, so when you present the paper, again, don't think about the information presented in the paper. Always try to have critical thinking. Why we are doing this way? Why it's only limited to 16-bit? What happened with the 4-bit? Is it the fastest performance? There could be some outliers with that scale. It's not always increasing, it's not always decreasing. Okay, so try to fortify all those sides in your presentation. Try to always ask yourself why we are doing it this way, but not another way. All right, so now we will talk about how enable complex operation in 3D stack memories. This is very similar to Ploto. That's why I try to cover it. Try to cover Ploto again quickly, just to give you some reminder. You'll see like really sharing large similarity with the Ploto, although the device that they are targeting is totally different. So here we are targeting 3D stack memory. Over there, we were targeting traditional DIMMs. Okay. Is it recording? I think it is, right? Is it recording? Yeah, I think it's recording. Let's verify. Yeah. All right. So now, because the paper that we are covering try to accelerate something related to bioinformatics. So we have to give you some solid background about the topic, and then we will move to the paper. Okay. So I'll try to give you a brief introduction to genome analysis. How many of you hear about genome analysis or bioinformatics? or read mapping. 1, 2, 3, 4. Okay. Yeah, we don't require that. So let's see what it's about. Initially, when we talk about genome analysis, we expect to analyze the genome, right? And the genome exists in our bodies, in any living bodies like viruses, bacteria, also in plants, trees, animals, and so on, right? So all of them have genomic data. So ideally, we would like to get a genomic sample, whatever it is, okay? And we would like to convert into text. That is your complete DNA sequence. Unfortunately, since the first try to do it in 1977 until today, we cannot do this process. Okay, we cannot send a genomic sample to anywhere, machine, black box, technology, technique, and we get the full sequence of our DNA. What we have instead as a workaround to get around this problem, because this is really something fundamental problem related to the chemistry that we have in the DNA sample. We got that sample, we try to process it in a wet lab. So there is literally a technician sitting in the lab and adding all those molecules together, trying to break down or chop your DNA into segments. And why we do that? We wanted the full sequence of your DNA, but we intentionally try to break down your DNA. Why we are doing it? We discussed a lot of things about those in the previous semester in advanced bioinformatics course, but I'm going to give you quickly the reason for that. So there are enzymes responsible or trying to help or facilitate the process of reading these segments, and those enzymes have a lifetime. Okay, so their life cycle quickly expire after short time. So they do small job and then they get tired. Okay, so we could preventing chopping the DNA, but we will lose that enzyme. Okay, so start reading some character from the DNA or some chemical molecules, and then the enzyme just expired. Okay, just stop working, and then we lose that DNA segment totally for good. So to leverage like large quantity or to make the sequencing really high throughput, like sequencing large quantity of those DNA, so we intentionally break down the DNA into pieces that is equivalent to the enzyme that we are using in the sequencing process. And when we say sequencing, meaning that reading the genomic material or converting it into a text. So the genomic material, this is your DNA, it's double helix structure, right? And at each point, we have a chemical molecule. At any point in this chain, we have chemical molecules. We discussed last time that those options are 4, okay? Either A or C or G or T. And those real chemical molecules, so each of them a molecule by itself. This is adenine, cytosine, guanine, and thymine, okay? Now to build this, when we look into any of those, we call it as a strand, it's really double structure again. So when we look into one of them, it's double structure such that whenever we have A in one side, we have the T in the other side. When we have the C, we have the G at the other side, and vice versa. Okay? This is how this is really tightly bonding to each other. There is a chemical bonds between those molecules to attach them or keep them intact all the time. Okay? So now our goal is just to read this complete sequence, just to identify whether at this point we have A, at that point we have C, at this point we have G, and so on. Why we do this process? This is very useful to know that, for example, one son has a certain disease, the other son doesn't have it. So if you get the full sequence of your DNA, you compare it with another individual full sequence of DNA, you will probably identify some variation. There could be single character, for example, is different from this, another single character that's different by that. And then we could say, okay, probably any of those two variations, or one of them, or both of them, are causing that disease. So we could develop a drug that only handling those two regions in the DNA in order to cancel the effect of those two variations. This effect you could see it in the current generation, like it's already converted into protein, and the protein is causing some phenotypes that observable to you, or you may not see it at all, but through several generations it will pass by the DNA. Okay, you pass it to your son pass it to your grandson, and so on, until one of those generations got the disease. They already hold those genetic variation, but not necessarily having the disease. And when we talk about variation, it's not necessarily a disease. It could be just phenotype, like eye color, hair color. Okay. So hopefully this is clear why we chop the DNA into pieces. And this chopping process is really random. You have no control about it. The only control you could do is the length of those pieces. But which is coming after which, we don't know. Okay, so it's really randomly you are chopping the DNA by sometimes you apply a force to it. Sometimes you apply ultrasonic signal so that you shred it. Sometimes you move it or you steer it in very high speed so that you shred the DNA into random pieces. Okay. Now, after we do this, what we call as a library preparation process in the wet lab, we send it to a machine that is called sequencer, genome sequencer. This machine could be really expensive, like $1 million and fridge size, really huge machine. Could be also handheld device, like just smaller than the palm of your hand, okay? And can cost like $2,000 or so, especially for Oxford nanopore technology from UK. Now, the sequencing technology, we have 3 prominent sequencing technology, and each of them designing or building different machines. We will see a few examples, but we explain a lot about it in advanced bioinformatics course. These technologies give you different output. Some of them give you a picture. Remember, our goal is to convert the DNA itself into text, the full sequence of your DNA. But some of them really giving you a picture of your DNA. Why? Because they try to do chemical reaction to these segments, and each of those chemical reaction emitting a light. And they have a CMOS sensor try to capture that light. If there is a light, mean there is a chemical reaction. If there is a chemical reaction, we already know what base is this. Is it A? For example, if the DNA molecule having A at that location, and I add myself a thymine to the molecule, and there's a chemical reaction happening, I know thymine always attached to A. It does not attach to C, does not attach to G, does not attach to T. So if there is a light emitting inside the machine, I 100% know that there was an A in the DNA. So by capturing that light, I could identify the full sequence of those bases. Okay. Now, this is one of the technology. Other technology use really different mechanism. For example, the handheld device use electrical signal to pass through the DNA. And by having alteration to the phase of the signal itself, we could say something about the character that we have in the DNA. So each of those chemical molecules will affect the electrical signal passing through the DNA in different shape. So I will cause some distortion to the phase of the signal, and that distortion can be connected to each of those bases. Now, we do some computational step to convert whether it's images or video, or this, we call it a squiggle, which is electrical signal, right? Passing through the DNA, so we have distorted shape over there. We use a computational step called base calling. Base calling will convert all those raw sequencing data into the text format. That will be unique and will be standard for all sequencing technology. Whatever sequencing technology we are using, by the end of the day, we will get that full text file. Okay. And anyone working on bioinformatics normally start their job after base calling. So somewhere over here. That is what we call genome analysis. I'll give a few examples of genome analysis, but before that, remember that this is happening in wet lab, this is happening in dry lab. That division is really thin line, thin border. People in computational side could still jump into the wet lab, improve something over there, and vice versa. wet lab people could still jump into the dry lab and do some new algorithms over there. For example, we already developed many algorithms that try to improve inside the sequencing machine to improve those images or those squiggle signal themselves. So there is a lot of potential for computer scientists to improve in this area. This is an example of the famous machine in each of these technologies. This is PacBio generating this length of, you remember we chopped the pieces ourselves. That is the length of the chopped pieces, which will be also the length of the output, because we chop the DNA into like 1,000 long. Then when we read it, also the output will be 1,000 long. Okay, so those are the length of those pieces coming from the machine. This one is fridge size. This is also fridge size. This is benchtop, small, but this can fit about 48 of the devices I told you about that could fit it in your palm of hand. So that single device, you can repeat it 48 times and fit it in that machine. But they provide that USB device, small one, cost about $1,200, and with all the consumable, cost like roughly $3,000 per sample, just to read one genome. Sometimes that genome is long, you need maybe 4 times that cost. Yes. Yes. Please raise your voice. Yeah, by looking by naked eye, you could understand what's happening. It's basically just a sensor camera. That's an excellent question. There is a lot of work. Try to do that without doing base calling. So you really don't need to convert it into text to do that comparison. But remember, those images are individual pieces, meaning that every time we progress in the strand by one character, we take one picture. Every time we move from one character to another, we clean the surface of the sequencing, the machine by itself doing that. We don't have to intervene. So it's cleaning its surface and then adding a new molecule. For example, now I'm at C. I don't know it's already C, so I'm adding the 4 molecules one after another, and I'm watching which one is causing the light to be emitted. Okay, so I add A. A does not interact with C, so nothing will happen. I wait enough time, then I wash the surface. I clean the A that I added. Now I add C. C does not interact with C. Nothing happened. Clean the surface. Do it again with the G. G interacts with the C, so it will emit the light. Okay? This way I will take the picture. So I take it only for one character, but the good thing, as you can see, you have a lot of dots. So if it's taking a light from one character, why we have a lot of dots? These machines are doing really high throughput sequencing. So this is the surface of the machine, and it has something called a pore. It's like just a hole, okay? And in each hole, we have separate individual DNA strands, okay? One of those segments. There's another hole, one of those segments, another hole, one of those segments, one of those segments, one of those segments, and so on. And you have many of them. In reality, it's really not one segment. We duplicate in each hole, we duplicate one strand like 1,000 times. So we have many of them in the same hole, but all of them having the exact same copy. Okay, we just copy them. Why we copy them? This is just technical details. Like I don't want to go through it, but this is just like to emit enough light for the camera to be captured. Because the scale of your DNA, think about one cell in your body. Inside that cell, there are a lot of components, and one of them is the DNA. And the DNA is about 3.2 billion characters. 3.2 billion characters inside one cell. So you can imagine the scale that we are talking about. So every character is really small, or each of those chemical molecules is really small. But we try to replicate it many times just to emit enough light whenever we do the chemical reaction. So back to your question, can we process those pictures and infer something about the comparison? Of course, yes. Yes, of course, you can do that. You can do that. But yeah, it might not be straightforward to do it because you will lose the location on which pore you are handling. Okay. So by the end of the day, you'll just get genomic sequences, right? It does not tell you which genome sequence is coming from which of those location in the machine. The picture has that location or spatial location, spatial locality. For example, if this light is emitting over there, I know that at that particular location, there was this particular character, okay? In the next picture, in the same location, I know that this is the next character in that strand. In the next picture, I know that there is a light emitting because this. So going backward from text to the image, it's a bit tricky because I already lost this information. I never record which strand is coming from which location over there. Okay, but I can still use these images to say something about the comparison. Yeah, it could be if you do the sequencing sequentially, or you don't need to do it sequentially. Once you do the conversion from the color into this, you could read them sequentially over there. So you always store this as the first sequence over there, this is second, and so on. Exactly, yeah, you could go row-wise, column-wise, and this way you already maintain the information about the location of those. I like this discussion because now the trend is to avoid the base calling step. The base calling step is a really expensive one where we convert those row sequencing data into a text. And this is really not necessarily. So the goal of base calling is just to make a standard output out of different technologies. Because one of the technology will give you images, one of them movie, one of them just electrical signal. They try to make them unique so that bioinformatician, when they develop a tool, that tool might fit all technology. They don't have to focus on one of these technologies. But when you think about it, you really don't need to do that. You can directly process those row sequencing data. Okay. Yeah, so hopefully now you agree that regardless of the sequencing technology, until today, we're still getting randomized small fragments of our DNA. Until today, more than 80 years, we're trying to do sequencing for the whole genome and we cannot do it, okay? How to sequence the genome? I already give you a lot of insights. We maybe took about 15 minutes just about sequencing. But I like this discussion. I have no problem with extending it more. We discuss about different technologies. There's 1, 2, 3, 4, 5. So if you are really interested into this, check our advanced bioinformatics course last semester. You'll see the lectures, 4 of them just about sequencing. Okay, you'll see full package of lectures around that. So if you're interested in this topic, you can watch. There you'll see some set of slides on the wet lab process, some other set of the slides on the dry lab. Okay, so if you are computational, you'll see something. If you are from the wet lab or interested in the chemistry, how we do the sequencing, you'll see other side of that. All right, now back to the story of the paper that we will present today. So every piece we got from the sequencing technology, regardless the data that we are using, we call it as a read, okay? As a read. That is a read of your DNA. Now the problem is that we are getting those pieces as randomized fragment, just random pieces of your DNA, and we want to put them all together as one piece so that we can interpret your full DNA. I told you those pieces have no information at all about their location. Which piece is coming from which chromosome, from which location, which is coming after which, we don't know. So likewise, when you solve a puzzle, whenever you have puzzle pieces scattered around, you really need a reference picture so that you can stick those pieces all together. Okay. That reference picture, we call it as a reference genome. So we really need the complete genome before we start solving this puzzle. The complete genome is about 16 people living in New York. Okay, so they do depth study to a 16 individual to study their DNA and build the complete genome for them, and they use it as a reference for the entire world. And we have that reference already publicly available in NCBI NEH database called as reference genome. So you can search about it. We've been already improving that genome draft since 25 years. Until today, we are still improving that draft. Maybe last month, we closed maybe about 99% of that genome. We still have some small gaps, but we complete most of that draft. So it took us about 25 years to just build that draft that we can use as a reference picture. But assume we have it. Now it's easier for us to compare every piece with that picture and try to guess what is the location of those individual pieces, so such that you can build back your genome or full DNA. You can think about it as really a text that you print in a letter or A4 pages and so on. And when you want to compare, yes? Yeah. And then we go back to point 0 and build back. Excellent question. We already know the size. It's 3.2 billion characters, right? And those sequencing machine, maybe I don't want to go back too much. Let's see how far. Yeah, those sequencing machine, they introduce their own error. Okay, they have their own error. So they might telling you that this is A, but in fact it's T. Okay, and sometimes they are telling you that this is C, but my confidence is 50%. It's up to you. Want to take it, take. They have that score. They record it in the file along with the text file. Okay, so they're telling you my confidence when I figure out what character it was there, 20%, 70%, 80%. Sometimes we don't consider those characters. If it's less than like 70%, we just record them as N character, meaning undefined, unbiased, whatever. Okay, meaning it's not among those chemical molecules, just artificial character. But when you open that human genome from NCBI, you can download about like 700 meg or so, but it's a compressed file. When you decompress, you will see about 3.2 gig because each character is really 1 byte. So 3.2 billion is really 3.2 gig bytes. When you open it, you'll see a lot of N's, especially in the beginning. You'll see really whole like 100,000 characters, just N's. That means this is a gap. We don't know what's the content of it. Good? All right, perfect. It's terrible. It's terrible. It's terrible that we are using that reference coming from 16 individual for the entire world. If you compare African genome to European, you'll see a whole bunch of differences. About 10%, normally African people having 10% more DNA bases. So it's no longer 3.2 billion characters, more than that, 10% more. Okay? When you talk about even cities inside the country, they may have their own different DNA structure. So it's terrible that we are comparing to someone else, but it's better than nothing. That's a great question. We don't know why. So NIH, when they started this project, it was, I think, 13 years from 2003. So it's about 1990, where they start that consortium as a human genome project. And it's not only about US, Japan was there, UK, Europe, all of them set together. And then they start for 13 years building the first draft. We got it in 2003. But even like last month, we got the most updated draft out of that. So we're still continuously building on top of that. Anyway, in that time, there's very famous newspaper who published the advertisement for volunteers. Anyone could volunteer their own genomic sample for that project. Okay, so among them, those 16 people donated their DNA. Now, why those 16? Why not more? We don't know. Like, it could be only those who agree, because they were something crazy. Nobody knows about genomics at that time. Nobody has the idea that this data will be used forever. Okay, and remember, it's not only about you. If you agree on donating your DNA, it means you are donating the entire family history for disease and so on. So they could really build your brother DNA, your mother, your father, and so on. Because one of those is coming from your father, one of them is coming from your mother. This is how you share the genetic material from your family. It's really good to be careful when you choose your wife or your husband. Okay, because that will stay forever through the entire generations. There is a very famous story. Yeah, we don't have time for all of these, but yeah, I'm happy to really extend the discussion. There's a very famous movie, even in Hollywood, about Henrietta Lacks. Or in short, it's known as HeLa. This is very famous. There is a movie about it. A very famous story. She had a cancer. She was in, I think, Johns Hopkins Hospital that time, and their doctor decided just to take some sample, some cells, the cancerous cell, and they kept it forever over there. They keep duplicating, sending to research institutes around the world. Anyone who wanted to study cancer, you can buy those cells from us. They duplicate them, send around the world. Okay, so after like 10 years, there are a lot of studies about rebuilding the DNA of the entire family of Henrietta Lacks. It's not only her. She already passed away. And until today, every once in a while, one from the family is suing that hospital. Every once in a while, every year, almost every 2 years. I am the 5th generation, and my DNA is already, without my consent, was leaked. And they closed the case like after a few years. They settled it down with a lot of $1 billion and so on. You'll see it in news. So just do the check. Okay. So it's really risky to donate your genetic material. But there are a lot of studies on how you could conceal the DNA or hide or add noise to the data such that you cover some particular sensitive information, and the rest of the DNA you still can donate it, for example. All right. This is how it looks like from inside. Really, when you open those files, you'll see just a bunch of 4 molecules. But remember, there are some other information about the confidence of the machine. So it will tell you for that character, for example, the confidence 70, this is 20, this is 30, and so on. Now, when we talk about workload, how many of you know this building? You spend maybe 5 years of your life in this building, right? This is 25 Park Place, right? How tall is it? 28 floors, maybe with the basement and maybe with the rooftop. It's about 30 floors or so, maybe more, right? It's already tall enough. Now, if I ask you to go to the department, download that file, 3.2 gigabyte, the reference genome, and print it on A4 pages or letter pages with small, very small margin, no line spacing, a very small font size like 11 or 10. How many pages do you need to print such amount? So you're going to print and stack all these paper as a pile, and you compare it to that tower. How many of you say it will be shorter than tower? How many of you say it's longer than the tower? Okay. How many of you say it's really same length, same height? Shorter? Close to it? Okay, let's see. How about this? Still getting some insights? Yeah, this is in math. This exactly. No, it's not close. There's 100 feet difference. Yeah, so it's really amazing. And that we are talking about single cell, single genome, single individual. It's really amazing how we could fit all this information in one cell. It's amazing. Now, when we talk about like thousands of people, that's a huge workload to be processed by today's computer, right? Okay, so now let's talk more about the biological side or biomedical side, okay, apart from the data itself. Now, assume we already read all those complete genome or the pieces coming from the machine. What could be the use cases? What could be the benefits of doing so? One of the cases, which is very famous, that we could stack all those genomes, right? And we stack them with the same length based on some similarity between the sequences. We could know that this should start at this location, this location, this location. And those are like 16 individual. So each of the lines over there is a snap of a genome for different individual, okay? And our goal is just to try to find the genetic causes for certain disease. So we stack all those genomic sequences on top of each other. We try to compare where they are similar, where they are different. When we see the black character, it means they are always the same content, nothing interesting. But when we look at the differences, we see like two columns, 1 and 2. We call it as a SNP. SNP is single nucleotide polymorphism, meaning that just single variation, okay? Just single character change from one individual to another. And we see there are two of those SNPs, one over here, one over there. And if we know that those individual, half of them as a case and half of them as a control, meaning that half of them have a disease, half of them, they don't have that disease. They are healthy from that disease, okay? Now we try to compare those DNA and figure out when those having the disease having a certain genetic variation, and when those not having the disease having something else in that genetic variation causing them to be healthy. Now, because we are smart people, here are a blood pressure reading. So all of you can do that at home, right? You can read the blood pressure and take the reading. Now we want to correlate what is the SNP that cause high blood pressure disease. What variation cause that those individual to have high blood pressure? Just look by looking to the data itself. Who says 1? Who says 2? And if you say any of those, which character exactly is causing the high blood pressure? C from 1 or 2? SNP 1? C from 1. Who says G? C and G. C and A or C and G? Okay. Who else? G and A. C as common and G and A over there. But we also have G and A everywhere. But C not. Okay. G is the main cause. G and C. What else? Yeah, I like this kind of discussion. It's really showing that you are interacting with the topic. Yeah, normally we are looking in those kind of disease for single variation, single cause for the disease, which is apparently C can be really correlated with the high blood pressure. And whenever we talk about low blood pressure is T. And the other things here, G and A is really random, just scattered around, has no effect. Because we have A's everywhere, we have G everywhere. Okay, so that's the answer. Who deserves the chocolate? Jahin. Maybe someone else? Who? You choose. Yeah, I'm going back with no chocolate. You'll see, okay. I like that. Thank you. Okay. Yeah, so this is a really real use case. This is not cartoon example. This is a real use case, and we use it in life. And this is called GWAS studies. This is very famous studies in bioinformatics. We do this for large population. So we don't consider 16 individual. The larger the sample that we collect, the better the accuracy. And normally we use thousands of those individual as control, 1,000 as cases. Cases mean directly or control a group of people having the disease. The other group does not have that disease. They could have something else as a disease, but the disease that we are studying, they don't have it. And then we compare and we build something, we call it Manhattan plot. And as you can tell from the name, like New York, it's full of those Manhattan towers, right? That's why we call it this name. And you can see all those towers in the plot. In the x-axis, we have all the chromosomes of a human, like chromosome 1, 2, 3, all the way to 23, okay? And the y-axis, we have the frequency, or what we call as significance, which is minus log the base 10 of the p-value. The p-value is related to how many times we saw that single snap appearing on those cases, okay? So we correlate that, and whenever we have higher frequency, like this snap is really happening often at that location between all individual, meaning that all those snaps above a threshold having something correlated to that disease. Which one of them, we don't know. Because again, in the previous example, it was easy because we are focusing on very small region in the genome. But over here, we are focusing on the entire human genome, the 3.2 billion. So we see a lot of those variation. There is a paper about TWAS, something else, transcriptomics, not necessarily the genomics. So if you are interested in this topic, read it. But something simpler you can do is to go over this database yourself. You can open it now, you can open it later at home, called OpenSnap. It's for free, publicly available, OpenSnap. And as you can tell from the name, this is a database storing all the snaps that we know. And they store the disease that is caused by those snaps, okay? Not necessarily the disease, sometimes the interaction with the drugs is also different because of that snap. So it's not necessarily causing the disease, but when you take a medication and your brother is taking the same medication, both of you having the same disease, you might getting better with the medication, but your brother is not getting better. You can imagine as complex as that. So that snap is causing one of you to get better by that drug, but the other one is not getting better, just because having different snap. So once you go to that database, you go to data, you choose snaps, you'll see something like that. So you can randomly choose any snap. You will get like a table of snaps. You can just click any one of them, or you can search for certain snap or certain disease if you're interested in knowing something about it. This is a unique name about the snap itself. This is the location happening chromosome 19. This is the exact location in that chromosome. And here you will see something interesting. I was telling you about this structure of the DNA, right? So if you are getting from your father a T, and you are getting from your mother T also in that particular location in chromosome 19, meaning that that individual having a probability of 20 to 25% of such hepatitis C patient respond to treatment. So you have very low probability to get treated by that medication if you have hepatitis C, okay? But now if you are getting C from your mother's side, C from your father's side, then about 80% to respond to that treatment. So your chances to getting better is much higher if you have that content in your DNA at that location. One coming from the mother's side, one from the father's side, and so on, okay? All right, so now let's talk a little bit. Until now, we didn't start the paper itself. We're just introducing or setting the stage. Now we talk about solving that puzzle. We have those individual pieces of the puzzle, and we compare to the reference picture to solve that complete puzzle, okay? How we do it? I told you that we pick every puzzle piece and try to compare to the reference picture. But in reality, you could do the following. You bring that blue piece, which is the read coming from the sequencing machine, and you compare it to the complete sequence that we have in NCBI database, which is the one with 16 individuals from New York City. Okay. So every time you keep moving and compare. Whenever you see high similarity, so probably that piece is coming from that location. This is what we call as a brute force algorithm, because we are trying every single option in the genome. So every single location, we are trying to check the similarity. Do we have similarity with that piece or not? If we have, yeah, tell me the exact similarity. What are the characters that they are similar? What are the characters that they are different? So this is a very expensive way of doing it, and cost us about MNK. M is the length of the blue piece, the N is the entire length of the red piece, and K, how many times we are doing this process, or the number of blue pieces, basically, which is the query. So if we have like million of those pieces, that K is not a variable, it's not a constant. It's already a significant portion of the equation. Okay? But if it is just 5 or 10, we can ignore it and consider it as a constant in the time complexity equation. That was a very expensive approach of doing so. How we do it in real life is not like that. So we have that read coming from the sequencing machine. We have that 3.2 billion character coming from the reference genome. We try to get a few markers in each of those. So we got few markers from the first piece. Here we check 3 markers. Markers mean just a subsequence. We use it as just identifier to tell us what are the potential list of location in the reference genome. I don't want to check every single location, but give me just few locations that I can start with. So I check here 3 pieces from the read, and I do the same from the reference genome. I use whatever mechanism. There are really large number of ways how you extract those pieces. So assume we got those pieces from the reference genome. The blue, of course, matching the blue, the red match the red, the green matching the green. And we do something like that, just matching all these pieces together. What do you think? Is this approach better than the previous one? Than checking every single location and see the similarity, or just having some markers between the 2 pieces and check where they match with the blue, with the red, with the green? I don't care about where the entire thing is matching. I care just about smaller subsequences, which is easier to search in the entire genome. Now, the only trick is once you identify those interconnected or disconnected pieces, is where should be the correct location to look at. So we got potential location over here. We have 3 pieces. We got another location over here, maybe 2 pieces. Another location here. Another location. I don't know if you consider this as one location or each of them another location. That's up to you. You can consider also this another location. This is 2 of them as one location. This is one location. But it might make more sense to consider this as the correct location. Why is that? Because the order of the pieces looks the same, and we have also the 3 pieces together, right? So we have the blue, the red, and the green following each other in the same order, and also we have 3 of them. Over here, we don't have the green, for example, and so on. So directly, using this heuristic, we could identify the potential location of that random piece to be located over there because they share the highest similarity with it. Now, you could ask me a lot of questions about where this approach might fail. For example, if there is a variation over here, if there's a snap over here happening, like one character is different, and we are looking for that exact subsequence to happen in the genome, we cannot find it, right? Then we are missing this blue piece, and again, we'll have only the location with 2 pieces, the red and the green. So it all depends about your threshold, how far you want to tolerate errors, and how far you want to go in depth and check like many of those subsequences to be shared between the 2 locations. We discuss a lot of this in the advanced bioinformatics, a lot of algorithms on how you extract all those information. So if you're interested, maybe you can take the course next semester. But anyway, that is a good introduction to the paper that we want to show today, because our goal in the paper is how we can narrow down or reduce the computation to identify the differences or the similarity between a piece and between the complete genome. So after we identify that location, we apply something like edit distance. How many of you are aware about edit distance? 1, 2, 3, 4, 5, 6, 7. This side, no, except Jahin. Half class, no. No edit distance. How about sequence alignment? Sequence alignment. About dynamic programming. How many of you are aware about dynamic programming? Don't get scared. There's no gun or something here. How many of you are aware about sequence alignment or similarity check? Any algorithm for checking the similarity between two sequences? When you do like Google search and you misspell one of the words, right? And you click search, then Google directly suggests something for you. Do you mean this, right? How do you do that? That is sequence similarity. So they check the similarity with every single word in their database, and they quickly telling you, oh, probably your word is very close to that word stored in the database, but that is the only difference you have. So do you mean this? Based on the edit distance. Edit distance mean how many differences between the two sequences. The smallest the edit distance is, the closer the two sequences are. Okay? How we do it? We do an approach called dynamic programming, meaning that we have a table where we record all values between the two sequences, and then based on those values, we do some computation to find out the number of those differences. I don't want to go through these details. It might not be relevant today. The paper is only relevant to this slide, so please remember what's happening over here, okay? So we're going to skip this. This is something about how we improve the computation over there. Instead of computing the entire table, the entire dynamic programming table, by the end of the day, we could only compute one diagonal in the table. That is only the things in the middle. Or we could compute something similar to this shape. Remember, this shape is part of the table. So the table is really actually like that. And we are computing only the main diagonal or a few diagonals around it. This is how we got the first shape. It's not really connected with the next shape. Each of those is really in separate table. Likewise with the second one, so it's in fact is really a table, and we start around the main diagonal. We have something like this, like bigger tree, and then we get smaller as we get closer to the final cell over there. Okay, each of those pieces is mimicking the number of cells that we are calculating in the table. You'll see a lot of those techniques again in the course, and these are the names of those. This is an exact pointer to where you can find those algorithm in the lecture. So if you're interested, you can check lecture 10 to 13. Now the very final slide about the introduction, what I wanted you to have as a takeaway is that sequence alignment is really expensive. Sequence alignment is a process where we identify the similarity, the differences between two sequences, okay? Because we are building that table, regardless if we are using any of those mechanisms to build only the things around the diagonal or the whole table, it's still extremely expensive because we take all the permutation between two sequences. For example, if there was an insertion or deletion in one of them, we try to shift this piece to the left, to the right, bring it a little bit one step, two step, three step, and try to check all permutation where it fits perfectly to the other sequence. That's why it's really expensive, because we are trying all those options. Now, to solve that, there is something called as a pre-alignment filtering. And we said this step is called alignment, sequence alignment, right? And we will say pre-alignment, it means a step coming before that step. And we call it a filter because try to filter something. So think about this query, and you find 3 locations in the genome, and you'd like to filter before you check or examine each of those pieces individually. So you always have 2 options whenever you check a sequence and another sequence. So either those sequences are similar to each other or not similar from each other, right? So either the 2 sequences are really similar or what we call dissimilar from each other. Now, what does that mean? What does similar here means, and what does dissimilar mean? Similar, the context here is really up to threshold. So if we set it like we said, the maximum we want in between any two sequences to be 4 variation or 4 differences, then anything below that number, we consider it as similar sequences. Okay? So if there are 2 sequences with 1 variation, then that is considered similar. 2 variation, considered similar. Now dissimilar, anything above 4, like 2 sequences with 6 variation. That's something irrelevant to us. Why we do that? Remember, we are studying disease here. And for most of disease, we know how many SNPs are there or relevant to that disease. This disease, for example, caused by single SNP, which is single variation, then it doesn't make sense to look for sequences that have 2 variation, for example. It's not relevant to us. So we want only to know either the 2 sequences are exactly the same or they have only 1 variation between them. Okay? So this case will be very useful to use this kind of filtering. So whenever we have dissimilar, meaning that they are really irrelevant to each other, so just filter them out and delete them. We don't want them. But if we find the similar sequences, those are very similar and very relevant and very useful. So we want to apply what we call sequence alignment on those pairs. Because sequence alignment will tell us where are those differences, like if 1 or 2, where are they? What type? Is it insertion, deletion? Is it just substitution, replacing those characters, and so on? So remember, those, again, they are irrelevant to each other. Irrelevant, meaning they are not interesting to the disease that we are studying. And any computation that we are performing on those irrelevant sequences, meaning that it's just waste of cycle. Imagine that we are running a huge algorithm to just compute something, and by the end of the day, we figure out that all the computation we did just trash. We don't want them because the 2 input sequences are irrelevant to each other, and we don't want them, basically. So that's why we call it an expensive sequences. So by the end of the day, we want to develop something that is a filter that directly detect if the 2 sequences are similar to each other or not similar. That is the goal of the paper that we will study today. You give me 2 sequences, I tell you directly if they have differences above certain threshold or lower than threshold. I don't care where they are. I don't care what type they are, like insertion, deletion, substitution, and all those questions. This question left to the next step, that is sequence alignment, which is the dynamic programming table. Finally, this is the paper, was published in 2018. I was part of it, called Graham filter. Graham filter, as you can tell from the name, it's really a filter that's filtering those sequences. Okay, so you have as input sequences, and directly your filter is just telling which sequences are similar, which sequences are expensive, so you can delete them or ignore them. Okay. The key observation, hopefully by now it's already clear to all of you. Data movement is expensive. It's really bad in FPGA, in GPU, and also in CPU. We saw a lot of example on moving data from one component to another in terms of time, in terms of energy. The key idea is to do this process inside the 3D stacked memory, and we got very good results compared to the CPU. We will see it in a few minutes. We will start with something called 3D stacked memory. What is it? 3D stacked, from the name, it's really stacked layers of DRAM. So we were talking about a 2D stacked memory, which is the DIMM itself. So you have multiple chips stacked next to each other in the DIMM, right? Now we have them really on top of each other. So the density now much higher, and all of them in one chip. So we no longer that we have that abstraction or division between the chips. We really have all those DRAM on top of each other in one package, in one chip. Now what is inside the chip? This is the whole chip. This is really one black piece that you can see in the computer or on the GPU or on the FPGAs. if you dissect inside it, you'll see something like that. You'll see layers of DRAM. So this is a whole DRAM. This is a whole DRAM, a whole DRAM, DRAM, and so on. And you'll see a logic layer over there. And sometimes that logic layer does not exist over there. It exists somewhere else. But at least what is common among all those architecture is that we have 3D stacked layers of DRAM. Okay? Now, the special thing about it, why we stack them inside the package rather than just having individual chip as we have in the DRAM. The interesting thing that we connect all those layers all together through something called TSV. This TSV, you can think about it as fiber optics, for example. And you know, hopefully, the difference between fiber optics internet and the traditional LAN connection between the computers. Do you remember the LAN? You're probably still using it in your offices in the university, right? To connect the computer. This is something really slow. But when you connect it through fiber optics, you can gain really high speed. And exactly the same thing over here. Remember, there's no data bus, like connecting the CPU to the HBM, for example, or connecting those layers of DRAM separately. In this way, we can really push for high throughput or high bandwidth data transfer between all those DRAM layers internally within the package itself. And this is possible because we are using over here something called interposer. So this interposer is really a silicon layer where you can have a lot of connection inside it. Do you remember the problem we said about the data bus getting really wider if you want to transfer like 100 bits all in parallel? So we use the SerDes to compress all those bits so that we can transfer it in one wire. Now with the exact same problem over there. We have a lot of wires connecting through all these DRAM on top of each other. Now how we could transfer large amount of data without using wider buses. So the interposer over there is really a silicon area, much better than PCB to have all those connections in parallel over there. There are different forms of doing it. One of it called 2.5D. It's not 3D, it's not 2D, right? It's not three-dimensional. It's just 2.5. Why 2.5? Because we slash something in it and we put it aside, which is the logic layer, where we compute, where we do the computation itself. So we have already the 3D stacked layer of DRAM, but the compute is somewhere else. But there is also real 3D memories where we have the compute underneath those 3D stacked layers. Apparently, what is the difference? Fundamental difference is the energy per bit. So this is picojoule. This is 12. This is 3.5. This is less than 1 picojoule per storing 1 bit. Okay, so we got better performance in terms of energy if we stack all of them in one package in one small chip, because we are not doing any off-chip communication. Sometimes the 2.5D is really useful because when you separate the compute layer or the logic layer from the memory, you can have many of those cubes. Remember, all of them just 3D stacked layer of DRAM. You can have many of them and only one logic layer because it's already isolated from the cube itself. But we still have everything in one package. It's not separate chips. All of them just the same chip, the same package. All of them integrated together. When we look inside, it's the exact same story as we have in DEM, because apparently those are 3D stacked layers of DRAM. So the exact same architecture, this exact same way how we access the data in the DRAM. Do you remember when I activate a row, then read, then pre-chart, right? And we could access only one chip at the time and only one bank at the time, but we could access all those chips in the same clock cycle by having shared data and control buses between all the chips, right? So the exact same thing over here, you can track it from the moment we send the command. This is for command bins. This is for the data bins in case we want to write or read through them. So we send the command through these several layers. So we have 4 layers over here. In one of the layers, we have the banks. So here you can see the bank. This is one of the sub-arrays. If you look into the sub-array carefully, you'll see exactly the same thing where we have the DRAM cells. We have the local sense amplifier, and we have the row decoder, so we can choose among those rows to be read. We also have the memory cell, exactly the same. So the exact same story over here. The only difference is number of channels and how many banks we can access. Now, because we don't have one DIMM, we don't have one channel, we have really several of them. Now, how we can access all those layers of memory? That is the only difference, or fundamentally different, between the DIMM and the 3D stacked memory itself. So if we want to access a row over there, we have to access through the chip over there, or let's say a layer of DRAM, right? So we got a 32 byte in one transfer instead of 8 bits. In the DRAM chip, we were able to only get 8 bits at the time, and we could get it from 8 chips, so we can combine them, and we have all the time 64 bits coming from the DRAM module in total, right? Over here, we have much higher number of bits that we can transfer per one clock cycle. We will see the exact number. So we transfer it from one bank over here. We activate the row, then transfer the data within the chip, which is the layer over here. Again, this is really not a chip because it's not isolated. So all of them in one package. Remember that. We transfer the data within a stack, meaning that within vertically, all the way to transfer to the interposer, where we have the silicon interconnect going all the way to the logic layer. Now, these are one of the configuration that you could have for the DRAM itself. So we have two options. This is real 3D stacked memory from Micron, one of the famous companies in producing those memories. You have two configurations, either 4 layers of DRAM or 8 layers. The exact same architecture for 4 is the same for 8. Doesn't have any real differences. The only difference is the capacity, how large the data can be stored in both configurations. When we check here, we have 4 layers of DRAM, and each layer has 2 channels, and we have several banks over here. We have 2 channel, 2 channel, 2 channel, 2 channels. So in total, it's about 8 channels. And remember, when we talk about channel, a channel is really enabling you to access data individually. So in parallel, we could access each channel separately, and we bring amount of bits that we will define later on, all in parallel. In DRAM, we have only one channel connected to the memory controller, so we can access only that channel to access 64 bits. Hopefully, remember those differences. Now, you could see here we have also a large number of channels, but in fact, they are still 8. So if you see the first layer is A and C, somewhere in the middle is also A and C, B and D, B and D, E and G, E and G, and so on. So it's still 8 channels, although we have 8 layers of DRAM, okay? So it's not 16 channels. The only difference that we could control or alternating between this layer and this layer at any time. So in this way, we can increase the size of the bank without increasing the complexity of the DRAM device itself. Now, what is the interface of the HBM? So we talk a lot about them being able to only transfer 64 bits at the time, or per clock cycle. How about the HBM? 1024 bits at a time. 1024 bits. Okay, 1024. And remember why we are able to have those many of wires. Imagine there are physically 1024 pins coming from the HBM. Because of the interposer, which is the silicon layer where we have all those wires going through without really seeing them physically. You're not able to see them as in PCB, for example. That's why we are replacing those connection between the logic layer with that layer of silicon, just to have that huge number of interconnects. And this really how you see it, regardless if this cube is really 8 layers or 4 layers, you still have all the time 8 channels. Each channel will give you 128 bits. And remember, you can access each of those channels individually, separately, and in parallel. That's how you get the 1024 bits at the time per clock cycle. 8 times 128, that will give you the 1024 bits. There is something critical about HBM. You could buy a really expensive one, and at the end of the day, it's giving you the same performance of DIMMs or DRAM. Why is that? Because of the way you access the data. And we saw that problem also happening in the DRAM itself. If you want to achieve the best bandwidth, you always have to achieve the data sequentially. Sequentially, meaning that you place your data in one bank and you keep accessing row by row, row by row. Now, if you want a little portion of that row and you keep jumping between the banks or you keep jumping between the layers in the stack, you are losing the bandwidth. Because remember, you access one layer, you go to a bank, you fetch the entire row. Now the row is much wider. It's no longer 8 bits. And you are physically transferring all those bits. And by the end of the day, you are using only a small portion of it. So you'll see really the performance or the throughput is dropping significantly if you go from less data that you would like to fetch and with irregular memory accesses. So this is very important if you want to deal with those kind of cubes. All right. Now, how you do the filtering? Hopefully, this will be a quick one because now I already give you the solid background for genomics. I already give you the background for those cubes. So let's see what is so special in this work. Now, the first thing, remember what's the problem that we are dealing with? We have a very long genome sequence, which is 3.2 billion character, and we have a very short sequences coming from the machine, which is 100 long sequences. So you have 100 long sequence, and you have 3.2 billion character long sequence. And your goal is to identify where that 100 character located in that long genome. Okay, so we draw a lot today. We have this short sequence and the very long sequence. We want to just identify whether that small piece is located here, here, or there, and so on. How we would do it? Let's see. Something called bins. This is invented in the paper itself. And how we do it? We partition the genome into large sequences called pins. So imagine this is the human genome, 3.2 billion character. What we do, we just extract overlapping or subsequences from that genome. Why is that? We will see. Represent each bin with a bit vector that holds the occurrences of all permutation of a small string. Sounds cryptic, right? But let's see what does it mean. So in that subsequence of the whole genome, we are going to look into sub-subsequences. Okay, in that subsequence, we are going to also extract sub-subsequences. What does that mean? Means we could bring the first 5 character, convert them into binary, and move on to the next 5 character, convert them into binary, and so on. Why we convert them to binary, we will see in a few minutes, but you could directly correlate it to the way we store inside the DRAM. Remember, in DRAM, only 1s and 0s, okay? And the granularity to access the entire row, then you can access columns. Please pay attention to that. How we will do it? We will build a vector that has fixed location for every single sub-subsequence that we extract. So if we extract 5 character, whenever it's, for example, all A's, we're always going to store it in the first element in the array. When it's different content, we are going to store it in different location in that array. So here's an example. Assuming here we choose the sub-subsequence length to be 5 character. So we are going to calculate all permutation. It doesn't matter if it exists in the subsequence or not. We're going to calculate all permutation. We start from 5 A's, then 4 A's T, 4 A's C, 4 A G, and then we keep going from 5 A's all the way to 5 T's. So all permutation that we could calculate. And every time any of those permutation exists in the subsequence, we record it as a 1. And anytime it does not exist, we record it as 0. Okay. Now, why we maintain all permutation? it's a waste of resource. For example, what would be the size of this vector if we are calculating all permutations? Yeah, it doesn't matter. But the location is the important thing. If it is second in the vector, it's different than the fifth. It's different than the sixth. Okay, but it doesn't matter that we have a lot of zeros. This is the goal, to have a way to tell us that this subsequence exists or does not exist. So by the end of the day, we're going to use this bit vector to represent that subsequence. We are converting all ACGTs into only that bit vector. So we ignore everything over here and we keep using that bit vector to represent that region in the genome. Now again, why we are doing all the permutation? Why not just calculating the subsequences and that's it? For example, maybe in this example, we have 1, 1, 1, 4 ones. So we only have 4 of those k-mers exist in that genome. Why we are calculating all permutations? So the first question, how many permutations do we have? We want to calculate all permutation of subsequence of length 5 and all of them. 2 to the power 4 to the power 5. 2 to the power 5. 5 characters, and calculate all permutation of those. So we start with all A's, then we replace the last character with T, for example, then we replace with C, with G, and so on. We keep going until we arrive all T's. Those all permutation of sequence of 5 characters. Sorry? N to the power 5. NB. What is N? What is B? 5 to the power 4. Okay. 5b to the power 4. No, it's already defined. Already everything defined. The length of the sequence, 5 characters. And we know at each location how many possible options to do, right? Because the DNA alphabet, just 4, either A or C or G or T. So what could be all permutation? How many of them? Okay. So what is the answer? In each character, we have 4 options. Excellent. What does that mean? Yeah, you are very close. So we have 5. We have 5 buckets. In each bucket, we could fit 4 options. And any option does not affect the next option. If we choose A in one of them, 4 times 4. Excellent. Excellent, both of you. So you understand what they said. 4 to the power 5, right? You can give it to someone else. So over here, either ACGT, 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 right? And any choice we made does not affect any other choices. So it's basically just 4 times 4 options times 4 options times 4 options times 4 options. That is 4 to the power 5. Okay? And that is really the size of the bet vector. So 4 to the power 5, that's the size of the bet vector representing any subsequence in the genome. All right, yeah, we do that. And we record 1 whenever that 5 character exists in that sequence. Do we have 5 A's? Yes, we have it over here. Do we have 4 A's and C? Let's see. Do we have it anywhere? No, probably we don't have 4 A's followed by C. So we record it as 0. And we keep doing that for the 4 to the power 5 options in the bit vector. And remember, we don't record those permutation in the bit vector. We just record 1 or 0, but each location directly represent those tokens. So this is really unique. Whenever I tell you that the first location in the bit vector has 1, you directly should know that there are 5 A's in the subsequence. Okay, that's why we do all permutation. If we didn't do the all permutation, then we are going to lose that information, because if I tell you the first location in the bit vector is 1, what does that mean? It means nothing, right? I cannot know if 5 A's exist or 4 A and C exist and so on. Imagine that I'm only recording the one with ones. So I'm just giving you the bit vector, something like that. What does it mean? Nothing. Nothing. Cannot correlate it to which subsequence I'm connecting, right? But now, since I'm calculating all permutation, so that bit vector has a special meaning about every single location in it. And we do that for the entire genome. So every time I extract overlapping bin, and that bin I convert into a bit vector, and that bit vector I keep it aside. Okay, I store it somewhere. I'm going to reuse it. I will ignore everything about the genome and just using those bit vectors. This is how it works again. So you have that snippet of the genome. We extract it as a bin. We extract the bit vector. We do all permutation. And then we start extracting one, another one, another one, another one. And we store all these ones and zeros. Okay? You could initialize all the entire bit vector with zeros, and then you could record only the ones later on. Now we will have the first bin, the second bin, the third bin, the fourth bin. What we are going to do with them? Remember, the problem was that you have a short sequence and you want to compare it with every single location in the genome and try to identify whether this is similar or not similar. So now I'm replacing the entire data structure over here with a set of bins. So now it makes sense to convert also the read into that bin. So we convert the read into the bit vector. And now every time I bring one bit vector from there and all the billions bit vectors I have there and try to compare with them one by one. Now, how we could compare, let's see. Yeah, here you'll see some statistics, which is important, about how many of those bins we could generate. You could correlate it to n, which is the length of the genome. And you'll see 4 to the power n times t. T is the number of bits over here, because that's the size of the bin. And n is the length of the genome. And y to the power of 4, because we have 4 options all the time. And that will give you really how many bits you want to record out of the entire genome. All right, so now this is how it works. So you understand how we convert the genome into all the bit vector. You do exactly the same thing to the read. You convert it to one bit vector. You could do multiple. If the read length is really long, then you could have overlapping subsequences and do the exact same thing to the genome. But this paper is from 2018. In that time, there was no long reads, all of them of length 100. So that's why we consider only pieces of length 100. There was no 150, there was no 200, and so on. In 2016, I think they start long reads, but was with accuracy of 70%. That's why it was not famous that time. All right, so we got the read. Now we extract all tokens, which are the subsequences of length 5. That 5 is not fixed. It's up to you to fix it. You could have 6, 7, 8, up to you. You know 6, 7, you know it? Okay, so you extract all the tokens. And you have the bit vector, one bit vector from the reference genome, right? You have many of them, many bit vector. You just bring one of them. You have all the tokens, and you know where each of them should be located in that bit vector. And now it's easy for you to compare 2-bit vectors together. For example, if I know there is one over here, what does it mean? No, if there is one in the bit vector, it means there were 5 A's in that location. Now, do I have 5 A's? No. It means that is the first difference I have between the 2-bit vectors, right? I keep doing that for the entire bit vector. And it doesn't make sense to look for the zeros. So I quickly can check the ones. Where are the ones in the bit vector? And I check, do they exist over there or not? If they do not exist, now I quantify. How many mismatches do I get? How many differences between the 2-bit vector? That number is really important because it's going to indicate the differences between the 2 sequences. I keep checking the corresponding one with the bit vector and then adding the how many times is similar or difference. And if it is less than a threshold, it means good. If it is greater than a threshold, mean too much differences, then you discard or you keep based on that decision. Okay? Is it clear? Very simple mechanism. You represent the huge genome using set of bit vectors, and you represent the read using 1-bit vector. Now it's easy to compare 1-bit vector for all sets of bit vectors. Now, how we do it in 3D stacked memory? This is something about how we set the threshold. Maybe we can skip, it's really not important, but you can think about when you extract all overlapping k-mers and there is one difference happening, it's going to affect all those overlapping k-mers. So you don't consider a single difference between the 2-bit vector as single difference between the 2 sequences. A single difference is going to affect at least 5 overlapping k-mers. So that's why you count every 5 differences between 2-bit vector as one difference between the actual 2 sequences. Because all those tokens are really overlapping. And you can see the example over here. This is a subsequence of length 7. And you can see one difference in the middle affecting all those overlapping sequences. You can do it yourself for one example, like all A's, replace one A with T. And you see now how many of those tokens overlapping will be shared between another copy without that variation. Now, this is how we do it in real life with ReadMapper. We can skip that part. There's nothing special about it. But those are the location coming from the genome. We send them to the filter. The filter will receive one read at the time, will convert into bit vector. The only important thing in this slide, that those bit vectors processed only once. The genome itself converted into bit vector happening only once because that genome is a reference. So it will be reused for any sample. Okay, everyone in the world is using that reference genome. So we do this pre-processing once. We store all these bit vectors somewhere. And every time we get a read, we just check all of them and see the similarity or the differences between the bit vectors. We can skip some of the nice properties. Now let's see how we implement it in real life in the 3D stacked memory itself. We have all those layers of DRAM. We have the TSV connecting all the layers. We have the logic layer. Remember, there is extremely high bandwidth between the layer of the DRAM. Now, the bank itself in each of the DRAM layer, it's about a matrix of DRAM cells. Now, either the bit vector itself, like it has this 4 or 5 to the power 4, which mean about 1,000 or so, maybe more than 1,000 bits or 4,000 bits. This 4,000 bits, either you can fit it in one row or you can fit it in one column. The choice is all yours. But remember, we cannot fetch columns from the DRAM. We have to fetch rows. Now, which setup is going to provide higher bandwidth or better speed in terms of checking multiple bit vector at the time? We saw the example of Pluto, if you still remember it. How many of you remember where we do the data layout of the lookup table in Pluto? So each lookup table was laid vertically in the DRAM cells, right? And we copy it multiple times such that every time we could access one row that represents one element in the lookup table. But we could check for all input vector, right? So that work, Ploto, was published way after the Grimm filter. So basically, Ploto was inspired by Grimm, not the opposite. But here, because we are explaining Grimm after Ploto, so Grimm, you can say like inspired by Ploto, okay? So we are going to do the exact same thing. We're going to lay out all those bit vectors vertically in the DRAM matrix itself, and we have multiple of them. So bit vector 1 in the first column, bit vector 2 in the second column, bit vector 3 in the third column, and so on. Now it will be something similar to this. So we have bin 1, 2, 3, 4, 5, unlimited ones, and we can access them row-wise. What does that mean? Means we will know if 5 A's exist in the first bin, or the second, or the third, or all of them, or some of them, right? That is in one shot. Just by accessing the entire row, we already got which of those bins have those 5 A's, right? Directly. Meaning that we are enriching the parallelism in the filtering itself. I'm checking with like 4,000 or whatever the length of that row, many bins in the same time. And this is really impossible to do with multi-threading in the CPU, for example. So you can think about the high parallelism that we achieve over there. Now, okay, where should I do the checking? So I can fetch that entire row to the row buffer. Now, what should I do? How would I compare it to the first element in the bin of the read? Remember, all those bins are coming from the reference genome. None of them is for the read. I just fetched one row representing the A's in which part of the genome. Now I have the corresponding bin from the read, and I check the first element in that bin. But where should I do that processing, basically, to compare that first element in the bin with all those bins in the DRAM? Yeah, where should I do it? I don't have any comparing mechanism in the HPM. Only I can fetch a row. I can read, write, activate, pre-charge. That is the DRAM. Remember, there is a logic layer. There is a logic layer in the HPM. Logic layer, you can really reconfigure to compute anything you like. Okay? In the Pluto, we don't have such things, so we invented our own match logic. We added to the DRAM itself. We add a processing element inside the DRAM cells itself to have a transistor, whether to allow the cell to open or close based on the matching logic. Over here, we don't have such thing, but we do have a huge logic layer that you can program for anything. And this is what we do. We propose our own comparator to compare that single element coming from the read with all comparators coming from the bins of the genome. So you can compare with like 4,000 elements right away in the same time, same clock cycle. And every time you access a row, you access all bins, you bring them to the logic layer, you have that bin from the read, you keep fetching the element and compare with 4,000 of them at the same time. And you keep progressing row by row, row by row. So basically, you sweep over all those rows to extract all the information. Now, every time you check that there is a hit, there is a hit, there is a hit, means all of those are matches. So you are accumulating the number of matches to know whether it's exceeding a threshold or still below a threshold. And this is how you filter, and you would know where are the potential location of that sequence in the genome. If it is similar to here, similar to there, similar to here, similar to this, and so on. If it is not similar to any of them, you will filter out all the bins, and you will identify only the similar ones. Hopefully, it's clear. Do you have any question? Any question? The order of them is really overlapping. You just extract all overlapping. the first 5 characters in the sequence, the very next 5 characters, which is skipping this character, and just read the next 5, read the next 5, read the next 5. And only those 5 will be recorded as 1 in the bit vector. But still, the entire bit vector is all permutation. It's 4,000 elements, regardless how many I extracted from the sequence. So those overlapping, I could extract only 4. But what I record is 4,000 bits. 4 of them is 1, and the rest zeros. Clear? And I do the exact same thing to the read and the reference genome. All right, you'll see here something about the overhead that we introduced. So we have about 4,000 incremental lookup table. Why we need lookup table? Store all the permutation, basically. Okay, or to extract the tokens and convert them into 1 and 0. Why we need 7-bit counter? For we are counting the matches, right? And every time we get a hit, we increase that counter. Because at the end of the day, we have to decide, is this number higher than threshold or lower than threshold? So we really need a counter to count all these ones, right? And the comparator, why we need it? Compare the bins, or the element in one bin and the entire row of all bins. This was really interesting paper. So it opened the way for accelerating genomics inside 3D stacked memory. It was really the first one to use the cube or the 3D stacked memory for genomic application. Normally, we see it for bitwise application or for like complex operation as in Pluto, which is general purpose. But this is the first time to see genomic application inside the 3D stacked memory. That's why it gained a lot of attention, a lot of citation, and so on. And after that, it enabled a lot of follow-up work on how to improve the genomic filtering inside the 3D stacked memory. You'll see a lot of more advanced paper after that with higher or better accuracy, better filtering accuracy. There are a lot of potential problems with GRIM filter. Can you identify one of those problems? I can grant you the 5 chocolate, all of them. What could be a problem with GRIM filter? I told you about the story of 2018. What was not there? Long reads. What's wrong with long reads? Why we could not use this for long reads? Think about the permutation that we do. Those tokens, what is wrong with them? Remember, the bit vector is only binary, 1 or 0. Excellent. Wow, you'll get all of them. Really, all of them. This is something really huge. This is a big problem because the long read could be a million long. It's not really 100 or 200 or 400. It's really a million long, right? And when we talk about 5 A's, how many times it will be repeated in that 5 or million long sequence? The chances that you'll face 5 A's over and over and over is really high. The high probability that those 5 A's will appear maybe 100 times. What if they appear 50 times in the read, but 100 times in the genome? How you could do the filtering? You just record either it exists or does not exist. But what if it exists 50 times here and 100 times over there? How you could say this is similar or not similar? You already lost that information. And that's the biggest problem with the GRIM filter. Now, if you want to tweak that, you could solve it by changing the bit vector. Rather than using binary information like 0 or 1, you could encode each of those cells into 8 bits, 16 bits. You are going, of course, to lose throughput because you no longer represent 1 column by 1 bins. You are representing maybe 16 columns by 1 bin, right? Because each element in the bit vector It's no longer called bit vector even. It's element vector or array, right? So each element now it's represented by 16 bits. So you divide your throughput by your speed, like how much you could filter, over 16. So you degrade down, but you still can support like much longer sequences to do the filtering. That was amazing. So yeah, thank you so much. Okay. Yeah, so there might be other problems as well. For example, the filtering accuracy itself, it's about how many of those k-mers appears, right? What if there's insertion, there's deletion, there's substitution, then you are going to lose those token. If there is any substitution in the middle, it's no longer exact match with those. That's another problem. And there are many of those examples you could figure out. You could really build on top of GRIM filter and do your own paper to improve it, whatever. For example, the HPM cube was used in 2018. It's already outdated. We have already more recent, more advanced cubes that you could leverage with different throughput, different logic layer capability, and so on. Okay. Yeah, that was it. Hopefully, we convey the message of GRIM filter in a good way. Hopefully, now you have a bit of introduction to bioinformatics. Don't forget, we are offering this course every other semester. So next semester will be advanced bioinformatics. The semester after, hopefully, will be advanced computer architecture and so on. So